There we go.
All right. Trick question. Everybody. Kyle, you do me a favor?
Yes.
Come on in the little walkway. Okay? 2 squats, 3 jumping jacks.
2 squats, 3 jumping jacks.
2 squats, 3 jumping jacks. And I'm sorry to the Vikings fan. Things are not going great. It's not pretty rough. It's pretty rough. So my neighbor's a Vikings fan. We talk about it. It's always rough. All right. Squat depth isn't great. All right. Now, here's the question. Okay. He just did this. Where did he get his energy? Okay, yes, maybe. Well, that's a good question. Beyond that, where did he get his energy from? What metabolic pathways or pathway did he derive that energy to do 3 squats, 2 squats, 3 jumping? Where did it come from? Creatine phosphate. Okay, you guys agree? Okay, very recent. Any place else? Was that it? Who thinks that's it? Okay. Isaac's the only bold one. Okay. The rest of you think not. So here's the fun part, right? Sarah, where else does he get energy from? If you're not thinking it's just creatine phosphate.
So like the one that's in the muscle.
The one that's in the muscle. Ah, okay, stored ATP. Okay, yes. Okay, good. That's good. So stored ATP, you got to use that before we do creatine phosphate. Okay. Anything else? Glycolysis and glucose. Okay. That it? Yeah.
Didn't you say last time that like all pathways?
Yes. All pathways. Thank you, Charlie. You mixed it down. I promise you this will show up on the test. Okay? All pathways, all substrates, all the time. Even if it's a very short-term thing, even if it is a very high-intensity thing, all the pathways, all the substrates, all the time. I suppose technically, once you've used once you're like 30 seconds in, you're probably not using stored ATP anymore. You really don't have an ability to resynthesize creatine phosphate. But at the beginning, it's all of them. Okay? All of them. We have to understand that. Okay? Now, for what Skylar did, which one was the predominant pathway? Where was he getting the most energy for doing that short-term thing? Where was he getting the most? Good. Okay. How much longer could he have gone before he likely ran out of creatine phosphate? I wasn't keeping track of time. How long you want to tell you? 10 seconds? How much longer could he have gone? Maybe another 5 or 10 seconds. Okay. I don't know. He was kind of cheating on his squats. His jumping jacks were really half-hearted in that way. So maybe we weren't using all of it. Could have gone a little bit longer. Okay. But that's the big concept, guys. Okay. That's the big picture. Even though we're going through all of these pathways, that's the big picture. We're going to use all of these pathways most of the time. And what we're trying to figure out, though, is which one gives us the largest portion of the energy that we need for a given activity at a particular instant in time. Okay? And so when it's short-term stuff like that, we're going to use our immediate system. Those are going to be the predominant systems. stored ATP, creatine phosphate, a little bit of monokinase, right? If he had kept doing that though, for another minute, right? Scott, are you done jumping jacks for another minute?
Good, but I don't want to.
Okay, I'm not asking you to do it. It's not a qualitative thing, right? We're not assigning a value to it. But if you could have, then we would likely have shifted. The immediate systems were gone and depleted. And so we're going to start ramping up what happens here with glycolysis. And then we're going to try to find a way to figure out what happens down here at the end of all of this. Okay? That's what we're working with. It's about supply and demand. We left off with this the other day. We left off talking about glycolysis. And so remember, glycolysis is the breakdown of glucose, okay, technically all the way to lactic acid. It doesn't always go to lactic acid. So what I want you guys to know when we talk about glycolysis really is glucose to pyruvate. Okay, glucose to pyruvate. A 6-carbon molecule is going to get broken down into 2 3-carbon molecules. Okay. As we do that, we put some ATP in at the start. We put in 1 or 2 ATPs to start, and then we get 4 back directly. Okay. I should mention this term. It's a chemistry term. It's called substrate phosphorylation, which is different than something called oxidative phosphorylation, which is what we're going to talk about next with the electron transport chain. So creatine phosphate and its breakdown results in substrate phosphorylation. You're going from one molecule, pulling the phosphate off, putting it directly onto our ADP. That's substrate phosphorylation. Oxidative phosphorylation is the deal where we ping our electrons down, we create a concentration gradient with hydrogen, and then we release the hydrogen back, and that drives an enzyme that's called oxidative phosphorylation. So we're going to make 4 ATP via substrate phosphorylation. We're either going to put in 1 or 2. Okay? We put in 2 if the glucose comes from the bloodstream. So if it's glucose that you've eaten, that's in your bloodstream, or it's glucose that's come from the breakdown of muscle glycogen. We do 2 ATPs in, we make 4, so we net 2. We net 2. If it's from muscle glycogen, we skip the first step. We don't need to use the hexokinase step. We put 1 in here, we net 3, because we make 4, we put 1 in. Okay? This is why during exercise, especially high intensity exercise, preferentially, we will use glucose that comes from muscle glycogen put into this process. Okay? It doesn't cost us anything really to break it down. It's faster because it's in much more close proximity to glycolysis, and we make an extra ATP. Okay? You guys see why that would be advantageous? Okay, good. Remember, phosphofructokinase, or PFK, is the slowest enzyme. It's the slowest step in this process. That's the rate limiting step. Okay? That's the rate limiting step. You're going to see in the things that you get on the study guide and on the short answer questions, asking you things about rate limiting step, what's the slowest step in each one of our reactions. Okay? And that's going to be that. Okay.
You said that glucose from muscle glycogen is better for infants, or?
So it's not. Well, I probably did not use the most precise term. Preferentially, we get more ATP by using glucose that comes from muscle glycogen than from glucose that's in the blood. And during high intensity exercise, that becomes a bigger deal because we need more energy and we need it right now. Okay? We need more energy and we need it right now. When I'm walking and I'm just kind of sitting here, that extra 1 ATP isn't going to make a huge amount of difference. I'm usually pretty good at being able to match supply and demand. And it gets really high. We're fighting for every extra ATP that we can get, so the muscle glycogen becomes the best place to get it. If we want to derive stuff in muscles. Okay, so last little piece here, and this is very, very important. Okay, and again, I apologize. I know chemistry is not most people's favorite thing, but as we're breaking glucose down, okay, we are undergoing a process that's called oxidation. Generally, with oxidation, what's happening is we're taking electrons away from something. We're oxidizing glucose down into pyruvate. Well, at this step, after it gets initially split into these 2,3 carbon molecules, we are going to peel 2 electrons off. Okay, we're going to peel some electrons off of this molecule, and we're going to bind them onto an electron carrier called NAD. Okay, and so then NAD is very positively charged. It becomes NADH. The electrons come, and then a hydrogen, which is basically just a proton, a hydrogen follows, and we now have NADH. Okay, so we're going to, for each of our 3 carbon molecules, we're going to reduce. When we add electrons, something we call that a reduction reaction. We reduce it, and we go from 2 NAD to 2 NADH. Okay. This step is vitally important. Okay, vitally important. We can do one of 2 things with this NADH molecule that I'm going to talk about in the next couple of slides. Okay, but if we're doing our accounting, glucose, 2 pyruvates, that's what we've made. We've made 2 pyruvates, we've made either 2 or 3 ATP, and we've made 2 NADH molecules. That's what we're getting from the Embden-Meyerhoff pathways. Okay. Okay. Now, I'm going to say this at the bottom, and it's an incorrect statement. Okay. But to me, it's the easiest way for it to make sense for you guys, probably given how you guys have learned about all of this previously. Okay. So what we're going to talk about now is what is the fate of pyruvate? Okay. Where is the pyruvate going to go? And this is why purely glycolysis is this first one, okay? Which some textbooks, some of you guys may have learned it as anaerobic glycolysis. I would argue that is technically incorrect. Every step in glycolysis is anaerobic. All we're talking about is the fate of the pyruvate. But pyruvate, making it into lactic acid or lactate, okay? So we get the pyruvate, and then the pyruvate comes into contact with an enzyme I want you guys to know called lactate dehydrogenase, or LDH. Okay? It comes into contact with LDH, and that is going to then convert the pyruvate into lactate. And in doing so, it's going to take the electrons from that NADH molecule that we made, and it's going to take them back. And it's going to hook them back on as we go from pyruvate to lactate. We're going to add some electrons back in. Okay? So in doing this reaction, we make lactic acid, and then we turn NADH back into NAD. This is important, okay? This is important because, let me go back to here, okay? If there are not NAD molecules available, okay? Think of an NAD like if it's an Uber, okay? If there's no Ubers available to take our electrons, then everything piles up right here, right? We can't proceed if there are not NAD molecules available. And so one of the things that converting pyruvate to lactic acid allows us to do is it recycles the NADH back to NAD so that it can come back up to this step in this whole process. Okay? That's really, really vitally important. Okay. The problem with doing this is we make lactic acid. And it dissociates into lactated hydrogen, and the hydrogen is your drunk friend who causes all kinds of problems no matter what happens, right? It inhibits everything. The hydrogen inhibits PFK and shuts down glycolysis. As I've said, the hydrogen inhibits calcium release from sarcoplasmic reticulum. It inhibits the formation of cross bridges. In addition to all of that, it hurts. It makes your muscles burn, so you want to stop. Okay? So the problem with this is it allows this part of the reaction allows glycolysis to continue, which is good, and that will help us continue to derive energy. But we can only do so much of this before the entire thing gets shut down. Okay? That's why glycolysis can only be our predominant energy pathway it tends to be during very high intensity exercise for maybe 90 seconds to maybe 3.5 or 4 minutes in peak. Okay. It just can't go longer than that without sort of leading to this feedback inhibition. What's good because it lets us continue and it gets us energy, which we need, which evolutionarily might be fine. I got to run away from something for a minute or a couple of minutes, okay? But it's going to get feedback inhibition, okay? In some ways, the more preferable thing to do, if you want to continue to exercise for a longer period of time and you don't want your muscles to burn, is to take the pyruvate, okay? And rather than making lactic acid, we want to take the pyruvate, stick it into the mitochondria, put it into the Krebs cycle, okay? And then we're going to end up through the Krebs cycle reducing a whole lot more NAD into NADH and FAD into FADH, so we can make a lot of ATP, okay? So what really happens, the idea between aerobic and anaerobic, it's really, is there an anaerobic fate of pyruvate or is there an aerobic fate of pyruvate? Glycolysis itself is completely anaerobic. There's no oxygen in it, okay? The other thing that can happen is the NADH that we make during glycolysis can get passed into the mitochondria, and we can donate those electrons in the electron transport chain, and then free NAD molecules from the mitochondria can also get passed back out into the cytoplasm. So there's an ability for these things to go in sort of both directions, okay? The enzyme that drives pyruvate into acetylcoenzyme A and therefore into the Krebs cycle of the mitochondria is an enzyme called PDH, or pyruvate dehydrogenase. Okay? Pyruvate dehydrogenase. Maybe at the end we'll do a little sort of people with their eyes closed example. But the simple thing about all of this is we make a bunch of pyruvate and it's just floating around in the cell. Okay. And whether or not it goes into lactic acid or it goes into the mitochondria, becomes acetyl-CoA, goes into Krebs, is which enzyme does it bounce into more often? Okay. So we'll talk about how we can kind of shunt those things. Other thing to remember, you have different activities of LDH, of lactate dehydrogenase enzymes, based upon your fiber types. Fast twitch muscle has a more active LDH enzyme. Why fast twitch fibers are considered more glycolytic. In fast twitch fibers, more pyruvate will get turned into lactic acid per the amount of pyruvate made than in slow twitch fibers. Okay? That's important. It's one of the reasons why distance runners generally don't have a huge amount. The elite ones don't have a huge amount of type 2 much. And it's why you have more type 1s, you can conceivably be a better distance runner. Part of it is that. Okay. Okay. So all I'm saying, this slide, I do this sometimes, you guys want me to go back. This slide just sort of shows some of the mechanics of NAD to NADH, and then another one called FAD to FADH. NAD exists in the cytoplasm and in the mitochondria, so we do it during glycolysis and in the Krebs cycle. FAD generally is only going to be in the mitochondria. This is only going to work when we're going through the Krebs cycle. We're going to grab electrons, but the point is the same. As we break things down, we grab electrons off of that molecule as it gets broken down. We put them on these carriers, and then they're going to move them someplace else, and we're going to do something else with them later on. That's the big takeaway. Okay, that's what we're doing with those. Okay, so we've got this. I talked about that thing. All right, so before we jump into sort of aerobic metabolism a little bit more, I want to talk about a concept, okay, that relates to how much lactic acid we're making. So this is as good a place as any to put it in, okay? How many of you have ever exercised to the point where it felt like your muscles were burning? Almost all of you? Okay, do we need to get up and do that right now? No, wake everybody up a little bit? No? We don't want to just do, what if I said, I'm sorry, this is terrible of me. What if I said, whoever can wall sit the longest gets 5 bonus points on the next test? Like I'm winning. I did that once in class. I had people hold books in their hands, hold their arms out the longest. You would have seen some people fighting for stuff. So right now, okay, right now, once you guys sit here, anybody's muscles burning? Okay. As I walk right now, I'm walking, okay? Most of the time when you walk, muscles generally aren't burning, right? I need more energy to walk, but it's not really getting it. I'm not making a lot of lactic acid right now. We're going to reach a point, okay? And the point is really an exercise intensity. So it's really a force production kind of standpoint that we call lactate threshold. I can do a finger stick in all of you right now, get a drop of blood, and I can measure how much lactate is in your blood. Okay? It's usually under about what we call 2 millimole. Millimole is just a unit of measure per the amount of fluid that we have. Okay? It's a concentration. You start walking, it stays the same. You start jogging very, very slowly, it stays about the same. Okay? We ask my man Skylar to get back up and do jumping jacks for about 2 minutes, it's going to start to go up. Okay? If I ask you to just keep doing squats or wall sits, it's going to start to go up at some point. Okay? Lactate threshold represents the point at which the appearance of lactate in your blood increases above what its normal baseline levels are. And this is going to demark an exercise intensity at which anaerobic metabolism or glycolysis going to lactic acid is going to begin to ramp up, okay? And it may not demark that it's exactly the predominant energy pathway yet, but we're well on our way to getting it, okay? As lactate in your blood begins to increase, the pH in the muscles, the pH in the blood begins to fall, if it becomes acidic, and you're going to experience that as my muscles are burning. You're going to experience that as it's going to start to feel a little bit more difficult or more effortful exercise. Okay? That's what lactate threshold is. That's what it's going to be. We will get into when we talk about the pulmonary system, okay, and the cardiovascular system. You make some lactate, it goes into the blood, we move it around, we take it to a different tissue, we do some things with it. You drop your pH, you have an ability to try to buffer that pH, and there's some other things that are going on. But in its most basic, that's what this is going to be. Okay? Every one of you has a lactate threshold, and that lactate threshold occurs at some percentage of your maximal capacity to generate energy. So your maximal aerobic capacity to generate energy. if you're in really, really good shape to run or bike or something, right? Your lactate threshold may occur at 90% of your max, which means you can run or bike or whatever very, very close to your sort of maximal capacity to make energy without making a bunch of lactate. Got really, really good aerobic metabolism. All right. If you're a sluggo like me, it might be at 50% of maximum capacity. So for somebody like me, I walk up a flight of stairs, I'm over lactate threshold. Some of you that are in much better shape than me, you can go run a 6.5-minute mile or something and you're not over lactate threshold. Where your threshold occurs in relation to max has a very, very, very high correlation with the pace you can maintain or the work rate you can maintain without experiencing discomfort and without kind of getting tired or experiencing fatigue. We can train this. We can train this. Okay. One of my master's students works with the basketball team. He's an intern or the kind of the GA for their strength and conditioning program. And they go and they just run sprints. It's like sprint 100 meters, you get like 15 seconds of rest, sprint 100 meters, 15 seconds of rest, sprint 100 meters, right? It's terrible. It's terrible. What they're doing by doing all of that is they're really ramping up lactic acid production. And part of doing all of that, learn how to tolerate it better, the pain, but also you're training your metabolic systems to raise that lactate threshold. We'll get more into that when we do aerobic training adaptations. Okay, but that's what this is. This thing at the bottom called the lactate shuttle is really important. Okay, lactate is not a problem. Lactate is not the enemy. It's the hydrogen that's the enemy. Lactate is very simply a 3-carbon molecule. Okay, so I'll do my own sort of thing here, right? If I just squat, okay, and I sit like this, okay, I need to be able to sit back a little bit farther. It's hard to do this. Get my weight out of my toes and back over my heels. But if I sit like this, okay, metabolism is up. At some point, right about now, I'm starting to make more and more lactate, more and more lactic acid in my blood. Okay, it gets into my blood, and then it gets shuttled into the blood. Other muscle tissue, the brain, will take that lactate, pull it in. It's not a problem, and then it will stick it in the Krebs cycle and just break it down and use it for energy. Okay. When you stop exercising, you've done very high intensity exercise and you stop, lactate levels are high, and then it's going to sort of exponentially decrease back there. Because what you're doing is you're essentially shuttling that lactate back into tissues, treating it just like a pyruvate, just like a glucose, and you're going to break it down, and it's all gone. Okay? I'm going to ask a question, and then I'm going to duck. Okay? Why are muscles sore the day after exercise?
Isn't it because they're recovering from the care that you got during the exercise?
Very good. Okay. Thankfully, no one said lactic acid. Okay. Very common misconception. Why am I sore the day after, or 2 days, or 3 days, or 4 days after I worked out? People will say lactic acid. Nope, it's not lactic acid. Okay. It hurts during because of lactic acid. Afterwards, within about 10 minutes, all the lactic acid's gone. pH is back to normal, lactate's all gone. All gone. Okay. It's micro tears that make you sore. But if anybody asks, that's what's going on because of this capacity to shut it. Okay. So I make it and then I use it for energy kind of at some other point. Okay. That's lactate threshold. If we graph lactate threshold, this is a person on a bike, right? They're doing a Peloton workout or something. This is a thing that I hear that people do, right? You're at very low power outputs. We're doing a finger stick every minute. We're measuring lactate in your blood. Okay. If you want to do this, come by my lab. We can do it for you. But stays very, very constant up to a particular point, and then it begins to, as the power output increases, it begins to go up exponentially. Okay. The person's working pretty hard right now, would be my guess. but it's basically the same thing. Now it's up. This is very, very near max. And so once we get to this point, we're really beginning to ramp up glycolysis. It's getting to the point where we cannot take all of that pyruvate and put it into the Krebs cycle. Down here, we're in a pretty good balance where most of it's going to go into the Krebs cycle. Once we get to here, there's so much that we can't. We're going to start making more and more lactic acid, more and more lactate. Okay. Talked about this. We talked about that. This is a kind of example of what the shuttle looks like. Okay. So here's a fast twitch fiber. Here's a slow twitch fiber. You will note that we're breaking glycogen down. The glycogen goes through glycolysis. We make lactic acid. That lactate then can escape the muscle into the bloodstream. It can escape the muscle and go to an adjacent muscle fiber. And once it gets into the bloodstream, it may get taken up by the heart. It may get circulated back around into a different muscle fiber. But the end goal of all of this is that once we've made the lactate, it goes somewhere, and then it gets broken down put into the mitochondria, broken down in the Krebs cycle, just like it was a molecule of pyruvate or a molecule of. Okay. That's really all that I want you guys to know. Now, the thing on the very bottom, and you all are not responsible for this, but it says MCT1 and MCT4. These are things called monocarboxylate transporters. Okay. These are the molecules that grab the lactate and move it around. There are more monocarboxylate transporters in slow twitch muscle fibers than in fast twitch fibers. So people that have more slow twitch fibers are better able to shuttle lactate. And therefore they're better able to keep those lactate levels relatively low. Okay. That's what that is. But you guys are not going to be responsible for that. All I want you to know about the shuttling thing is when we make lactate, it's just a carbon like any other, and it's going to get shuttled around to other tissues and then broken down in aerobic metabolism. The lactate's not the enemy. Okay. Okay. Aerobic metabolism. So realistically, all that we have left to talk about is Krebs cycle, electron transport chain. Okay? You don't have to know all the steps in the Krebs cycle. You don't have to know all the cytochromes and things in the electron transport chain. But what I do want you to know is that aerobic metabolism may refer to glucose, okay, if it goes to pyruvate and then comes into the mitochondria, or fat. We only can break down fat in the mitochondria, so it can only be broken down aerobically, or potentially protein. And what we're going to do is we're going to take our pyruvate, or we're going to take our carbons off of fat or protein into the mitochondria. We're going to couple that with oxygen, and we're going to make CO2 and water and ATP. Okay? I'll show you where the CO2 comes from. I'll show you where the water comes from, and I'll show you why the oxygen is of 100% importance. This is, even during high intensity exercise, this is the primary energy pathway in your body. Okay? Because you can make so much, okay, even during very, very high intensity exercise, you're still going to get a bunch from aerobic metabolism. The issue with aerobic metabolism is you need a constant supply of carbons and you need a constant supply of oxygen. Okay? Which is good because most of you don't ever like hold your breath or walk. Oxygen is usually not a problem. Okay, we go to Mount Everest, then it may becomes a little bit of an issue, but generally that's not going to be a problem. Okay? Mostly it breaks down and we use fat, okay, and we use some sort of carbohydrate. When you're resting, it's a little bit more slanted towards fat. When you're exercising, it's a little bit more slanted towards carbohydrate, depending upon the intensity. If it's higher intensity exercise, it's going to be basically no fat and all carbs. Assuming we supply carbons into this and oxygen, our ability to make energy in aerobic metabolism is basically limitless. Okay? The system does not make anything that will inhibit any other part of the pathway. Unlike making lactic acid where we make hydrogen ions, unlike running out of creatine phosphate, there's no portion of this that leads to any kind of feedback inhibition. And as long as we've got glucose and fat, which in general we should have plenty of, and plenty of oxygen, we can just keep doing this. The issue is the maximum rate, and the max rate of doing this pathway is slower than creatine phosphate. It's slower than breaking down free ATP. It's slower than the myokinase because those are all one-step reactions. And it's slower than glycolysis because we basically have to run all but the last step of glycolysis just to get things ready to go in. Okay? So it's slower. And that's going to be the place where we're going to have a little bit of problems. Okay? It occurs in 3 basic phases inside the mitochondria. Okay? We make the pyruvate at the end of glycolysis, and then we have to shuttle the pyruvate into a mitochondria. It then gets converted to something called, I didn't spell it right, it's not actyl, but it's called acetyl-CoA. Acetyl-CoA. An acetyl group has 2 carbons. We lose a carbon as we do this, and it becomes carbon dioxide. The acetyl-CoA then goes into the Krebs cycle, which I'll show you guys some pictures of. You guys have probably seen 14 times. And then as we go through the Krebs cycle, we make a little bit of energy, we steal a bunch of electrons, we then take those electrons on NADH and FADH and go to the electron transport chain. Use the electrons in their kind of charge and making a hydrogen gradient to make ATP. So, what you see here is basically the transition step from pyruvate to acetyl-CoA at the top, and then the Krebs cycle, or the citric acid cycle, or the TCA cycle, or whatever you guys have learned it as before. Okay, so at the very, very top, you see there's pyruvate. Okay, there's some pyruvate. As we go from pyruvate to acetyl-CoA, pyruvate has 3 carbons. Acetyl-CoA, acetyl groups have 2 carbons. 1 carbon gets broken off, and it makes carbon dioxide. Okay. You will also note that as that happens, we're going to take some electrons, hook them onto NAD, and make NADH. Okay. So we've lost a carbon, we've made CO2, and we've also reduced an NADH. We've got electrons that we can use later. The acetyl-CoA then loses the coenzyme A. We hook acetyl onto something called oxaloacetate, which has 4 carbons. We make a 6-carbon molecule called citrate. Citrate then gets moved down and around and through the cycle, okay? And as it changes its shape, it's a couple of different steps. We're going to lose carbon, so it goes from being 6-carbon back over into a 4-carbon over here. The primary thing that I want you guys to know, we need to know the steps where it happens. It's just as we break citrate down, we are going to make carbon dioxide, and we're going to reduce NAD to NADH at these 2 steps, okay? Also over here, we're going to reduce FAD to FADH here, and we're going to make 1 molecule of ATP. So for every pyruvate that comes in, we make 3 molecules of carbon dioxide, okay? 4, we make 3 in Krebs, 1 up here, 4 molecules of NADH, and 1 molecule of FADH, and 1 ATP. Okay? That's what happens in Krebs. This enzyme here, isocitrate dehydrogenase, this is the rate limiting step. This is the slowest step, okay, in all of this. So what has happened now is we've made a little bit of ATP, but we have got a bunch of electrons that we can now take and use in the electron transport chain to make not just a little bit of ATP, but a shitload. Okay. And that's the really important thing that's happened here. But again, because there's all of these steps, it's slow. It's slow. Okay, so we've done that. I did that. Let's talk about the electron transport chain. Okay. So the Krebs cycle is taking place inside the mitochondria in the matrix, in the very, very middle. Okay. The electron transport chain is located on the inner membrane. You guys remember you got a mitochondria, and then it has an outer membrane, and then inside of it, it has this kind of inner fold of membrane that's going to be there. Well, in that sort of middle membrane is the electron transport chain. It is composed of things called cytochromes. Cytochromes are molecules that have iron in them. Iron is very, very strongly positively charged. Okay? Because it has such a strong positive charge, it wants those electrons. It needs those electrons, okay, that are on FADH and NADH. So what's going to happen is the electrons are going to be donated from these carrier molecules onto the cytochromes, and then they're just going to move from one cytochrome to the next, to the next, to the next. As that happens, okay, there's going to be all of these hydrogens that are going to be sitting around, because when the electrons leave, the hydrogens also leave. We move them down, and then we use the energy of them moving to pump hydrogens out of the inside of the mitochondria, sort of between the two membranes. Okay, it creates this big hydrogen concentration gradient, and then at the very, very end, there's an enzyme called ATP synthase. We let the hydrogens move back down their gradient. It drives ATP synthase, kind of like water driving a wheel, like a mill wheel or something, and then that makes ATP. Okay. And then oxygen, the electrons move all the way down our cytochromes, and then oxygen is where they're going to end up. We go from cytochrome to cytochrome to cytochrome to cytochrome. Then, at the very end, oxygen grabs them, and then the oxygen with the electrons grabs some of those hydrogens, and it makes water. Okay? So if you don't have oxygen, you cannot finish the electron transport chain. It will get stopped up. It will not function if you don't have oxygen at the very, very end. So, got a better picture here. Okay. Here's the mitochondria. Here's the outer membrane. This is the inside of a muscle cell here. Outer membrane, inner membrane, and then the inner compartment contains a thing that we all, the cytosol in here is called the matrix. Okay. And so what happens is, here's the NADH, here's the FADH that we made in the Krebs cycle, which is going on in here. The electrons come, they get donated. Here are our cytochromes, 1, 2, 3, and 4. They get donated on these. As the electrons go, then the hydrogens follow, and you accumulate all of these hydrogens out here between the inner and outer membrane. Okay. Electrons move, 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 move. And then here at the very end, this is the ATP synthase enzyme. It has a channel that will open. Hydrogens move back in from high concentration to low. That drives this enzyme, and then that enzyme will put phosphate onto ADP. It makes ATP. Okay. And that's how we're gonna run this whole thing. And then the electrons, once they get to here, are then going to get bound onto oxygen. They're going to grab these extra hydrogens and put it right back in, and we make water. That's how this whole pathway works. Okay. Things for you all to know. Chain is on the inner membrane. It has cytochromes. Cytochromes are strongly positively charged. So they really want those electrons. NAD and FADH donate the electrons. Pass down the chain. Creates the hydrogen gradient. And then we use that gradient to drive the ATP synthase enzyme. Oxygen is what we call a final electron acceptor. Then we make water. That's it. Okay. So if we do some accounting on all of this, we can make, the numbers seem to vary like every other day, right? Because some will be like, oh, with an NADH molecule, you can make 2.5 ATP. Well, you can't make half an ATP. You make 2 or you make 3, it doesn't really matter. But through all of this, if we count up everything from the start of a glucose molecule through glycolysis, through the intermediate step of going through acetyl-CoA, through Krebs, through to here, we can make somewhere between 36 and 39 ATP molecules, depending upon whose paper you want to believe. Okay? That is a huge amount more than you make through creatine phosphate or through the myokinase range. Okay? So while it takes a long time, it generates a tremendous amount of ATP. Tremendous amount. And it doesn't make anything that could either run out, like creatine phosphate, or that's going to cause feedback inhibition, like making lactic acid does. Okay. This is just basically an overview of what we're making in the electron transport chain. Okay. You can do sort of an ATP count and you can get there. I like this particular slide, and I'm not going to ask you guys to draw it, but I know that sometimes it's tricky to kind of picture how all of this looks and works in 3 dimensions. All right. And so what we can see is out here and here is the inside of a muscle fiber. In here is inside of a mitochondria that is in that muscle fiber. And so glycolysis, myokinase, creatine phosphate are all happening out here. Okay. But we're going to turn glucose into pyruvate. That pyruvate is then going to get made into acetyl-CoA. So you can see that the Krebs cycle and what we call oxidative phosphorylation of the electron transport chain is going to be in here. You can see the passing of our electrons potentially from glycolysis in, passing of electrons from Krebs into here, and we make our ATP. I also want to illustrate a pathway that we've not really talked about yet, and that's the breakdown of fat, and that's called beta oxidation. Okay, so beta oxidation, or breaking down of fat, only occurs in the mitochondria. We have to bring the fat into the muscle, and then we have to get it into the mitochondria. And there's sort of, it's going to make acetyl-CoA that will go into Krebs. It's just going to come from a different starting point. Okay. But I hope in some ways that this kind of gives you all a 3-dimensional sense of what is happening and where. Okay. And so remember that even as you guys are sitting there right now, all of this is happening simultaneously. You're making a little bit of lactic acid all the time. You're making some pyruvate that gets converted into acetyl-CoA and goes into the mitochondria all the time. You've got some fat, assuming you've eaten some level of fat at some point, or if you haven't eaten anything, you may have broken down adipose tissue. You've got some fat coming in. And the end game of all of this is just simply to either make ATP here and make ATP here, such that there's enough to do whatever it is that you need to be able to do. Okay? That's the end game. You just need enough ATP. Where can we get it from? So it's just that kind of net of everything that's going on simultaneously. Okay. So the last little thing we're going to talk about today before you guys can take your quiz, I'm going to start talking about the thing that we've kind of woven in and out the entire time, but it's called the temporal organization. This is the time organization of metabolism. Which pathway is active when? Which pathway predominates when? And that's dependent upon 2 things: energy demand and the speed of a given pathway. Okay, when demand is high, we need energy immediately. So we're going to try to get it from the fastest pathways. When demand is lower, then we're going to get it more so from our slower pathways. Okay. And so each pathway has its own rate control step that's going to help us determine how fast it can be and how we're able to best meet supply and demand. Okay. So all of them are active, but kind of which one contributes the most is going to come down to this. So remember, creatine phosphate's the fastest. Glycolysis, and by glycolysis, I mean making lactic acid from pyruvate is the second fastest. And then oxidative phosphorylation or aerobic metabolism through the electron transport chain is going to be the slowest. Right. Rate control of almost all of them is controlled by ADP levels. But in its most primary thing in creatine phosphate, the pathway is driven by ADP. Cellular ADP goes up, we immediately start breaking creatine phosphate down. One step, very simple, use it up until it's gone. Once it's gone until we stop exercising. That's what's going on there. Yeah, no, we'll save this for Monday. There's no sense in starting. I can't finish this in just a couple of minutes. Okay, so we're going to stop here. The quiz should be open at 9:05. Okay, at 9:05. Take the quiz. Let's see how this goes. I mean, once everybody is done, I'll give you your assignment for Monday. Anybody still working? No. Anybody doing it? Have anybody taken it twice? If you could. Okay, here's what I want. For I make this for Monday, or I make it for Friday? Monday? There's no football this weekend, right? Are we gonna get lots of studying done? Sure. Yes, lots of stuff. Okay. Everybody write this down. Here's what I want. Okay. I want you to write out each metabolic pathway that we've discussed so far. I want you to include the molecule that goes in and whatever comes out. And what comes out may only be ATP. What goes in, what comes out. I want you to include in that how much ATP per molecule that goes in we make. Also then want you to denote what the rate limiting step or part or enzyme or whatever for that particular pathway is. We got two more things. People want to take home assignments, so we're getting take home assignments. Okay? The other thing, this is the thought question. This is the thought question. Okay? If we understand what goes in and the rate limiting steps, how does exercise drive an increase in what goes in and in the speed of those pathways? You know that when you start to exercise, you need more ATP, and we are able to, for the most part, meet that demand. So there must be something happening with exercise that ramps up what goes through and ramps up how fast that they go. What is that? What are those things? Sure. And I probably can't. So, what is it about exercise that's going to lead to an increase in metabolism such that all of the things that go into these various pathways increase? And for some of them, it actually will allow the rate limiting steps to be sped up. Okay. That's the question. There is a right answer to that question. That's probably a graduate level question, but I want you guys to stretch. I want you to see what it is. Like, oh God, don't make us do this. That's where it is. Okay. Lastly, a fun one. Yep, fun one. I want all of you to write one short answer question and provide an answer that would cover something from resistance training or metabolism that might be a reasonable thing to ask on the test. Okay? I will get possible short answer questions to you guys today. I need to comb through the metabolism lecture and make sure I'm happy with what I wrote yesterday. But I will get those to you today. Okay, today. There was discussion on last time that having a study thing the day before. Some of you thought it was good, some of you were like, no, it was too close. It was too close to the thing. You guys want to try to do that again? And then if so, when? Monday? Yeah, Monday? Okay. Then we'll settle on a time then in class on Monday. Bring your answers to these questions and these things, bring it to class. Print it out or written down on a piece of paper on Monday. Okay? Thank you. Have a good weekend. Make good choices.